Mm-hmm. Okay, okay. Mm-hmm. Mm-hmm. ਤੇ ਐਗਜ਼ਾਮ ਕਿੰਨਾ ਔਖਾ ਹੁੰਦਾ ਹੈ? Mm-hmm. Okay. Mm-hmm. 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 Okay, okay. Mm-hmm. Mm-hmm. हाँ हेलो हेलो. हाँ सुन रहे हैं मैं. हेलो. मैं ਕਿਹਾ ਇੱਥੇ ਹੀ ਆ. ਭਰਾ ਸੁਣ ਸੁਣ ਮੇਰੀ ਨਾ ਕਲਾਸ ਚੱਲ ਰਹੀ ਹੈ ਹਲੇ. ਮੈਂ ਕਲਾਸ ਕਰਕੇ ਕਰਦਾ. So very few students have joined us in this time actually. I don't know, shall we continue the class? We wait until we have a number of students, though it will be recorded, as everyone agreed to that we can have the classes. So based on that, I just started this class. If there is any problem or they say no, it's not good, and we need to fix at 5 p.m. then. But I need 2 students to be available here. Hello, hello, hello Shipra, hello Rahul, hello Shwarya, hello Kunal, hello Smita, Gopika, Tanvi, Soumi, Urvi, Dr. Urvi. Yeah, from here we are done with the PCR and we're gonna start with this topic. So just waiting for few 4 minutes. Maybe just we wait for 4 minutes and then we start. Yeah, I shared these books earlier. So this is the best book actually, Concept of Molecular Biology. So I made this actually book by myself, and all the questions like significance of Chargaff rule, Watson and Crick model. So it's meant from the examination point of view. If you have any subjective exam, so you can write with the help of this. Michelson-Stahl experiments, Hershey and Chase experiments, and so on. Yeah, one of the best books ever. So it's made actually from the student orientation perspective. It also has all the transcription and translation and the within the translation steps. There are 3 steps: initiation, elongation, and termination. So they are also being explained very nicely with the full diagram and with the various steps in it. So one of the brightest books ever. You can use it for yourself. And some nice terms are coming up like various terms: RNase protection assay, PCR, RT-PCR, nested PCR, real-time PCR, southern blotting, northern blotting. So all with the statements, very nice. Your most of the topics are being covered in that. And also at the end you will have your MCQs, very nice, around how many? Around 84 MCQs with very nice from the all perspective, been touch in it, all the topics, good for the examination point of view also. So one of the best books ever, Concept of Molecular Biology. um, yeah. So hello, 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 hello. Hi, 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 everyone, everyone. So I have the audience here who really wants to study, who are committed to study things. So thank you very much for coming now. I hope it will be fine to continue the lectures. So we just, I make this presentation. So the PCA was over. So now let's start the session with one night session, late night session. Yeah, get yourself popcorns, some drinks, and enjoy this part. Don't lie in your bed with a blanket. No, no. Otherwise, you will sleep in the halfway. Be on your chair with the table. Keep your mobile or laptop in front of you. Yeah, have your copy and pen. Make it like a class. Yeah, not just to um, for the sake of taking the lecture. Right. So try to grab as much as you can from all these techniques. Believe me, once there are many students from mine who have covered this training. You can do your BTech for 4 years, you can do your BSc MSc for 5 years, you can do your MTech for 2 years, or even you can do your PhD for like 5 or 6 years. Maybe not everyone aware of these timings. That's true, Dr. Urvi, but I try to write um, I wrote in the group actually and asks everyone permission that if it is okay. And beyond that, I asked in my previous class in the evening that can I make, there were around 50 students in that class, and they all agreed to that. So on that note, I made this class at 9 p.m. also. So twice classes, two classes in a day. Maybe I should use these big emojis to ask for students. I don't know what to use. Hand gesture. So these things, if you want to have a career within the molecular biology, yeah, if you want to have a career within the molecular biology, or you know, biochemistry, molecular biology, and all corresponding allied fields. So the techniques that we're gonna discuss actually, they are meant to be from the job oriented perspective. Whether you work as a RA, whether you are an MBBS doctor, whether you are into R&D, whether you are a bachelor student, somewhere at the end of the day, people ask which techniques you know, because that will be, you will be working on the bench in the real life. Yeah. So in that sense, yeah, in that sense, we should um, make sure that all these techniques that we're gonna do here will be, you should absorb them like a sponge. Whether it's a Western blotting, whether it's a recombinant DNA technology, whether it's Southern blotting, or I don't know, we will deal with light microscopy and electron microscopy also after these, cell culture. So all these things are day-to-day life, and molecular biology and biochemistry techniques, it's a combination of let's say around 50 techniques. Yeah, first you work on the cell culture, then you work on the immunofluorescence, the light microscopy, then you work on the biochemistry part of it, then you work on the molecular biology part of it. So you can see within a 1 month or 2 month, while working on these techniques, you can bring a very big results, which might be difficult to understand for you at the moment. But if you are in part of research, if you have done that thing in your previous experiences, you will know how much important these things would be. And there are two things. There is a researcher and there is a technician. Yeah, there's a difference between researcher and technician. To a technician, if you give one protocol, he will read the protocol and will do as it is written and will give you the results. That's it. He will not see what kind of results you were expecting, why you got these kind of results within the cell, why what could be the reason behind that. But as a researcher, he will think all the perspective, yeah, so which a technician might be not. Technician work is to give you from A to B, but a researcher work is to go from A to B. How we can go from A to B if we are not able to go, how to do the troubleshooting, how to make things works. So by the end of the day, the question that you are looking for, the hypothesis that you are making, it will work for you. Yeah, so that's the main thing behind these techniques. Second, how accurate you are. Yeah, you're starting 8 a.m. in the morning for experiments, and you have to work till 5 p.m. You started with one experiment and somehow you are not successful with the results. You attempt it twice, again not working out, then you stop working about that. You don't want to concentrate on that, because time is the key in the field of research. If you're gonna spend your 1 month by repeating that experiment again and again, again and again, again and again, in order to get the results out of it, it will be just yeah, different way. Yeah, researcher field is part depending. So what steps you take, what makes you things will be ideal for you, it all dependent upon that. So be yeah, result oriented always, that how you can make the results best out of it. So macromolecule blotting and probing. So in general, this is the very layman or principle of all the blotting techniques, that you have a genomic DNA, then you cut them, restrict them or digest them with restriction endonuclease. Yeah, after restriction them with the endonuclease, um, you add your agarose gel electrophoresis. Yeah, you add your agarose gel electrophoresis. Then after adding to the gel, it will go through high molecular weight and low molecular weight under the positive or negative electrode. Then you transfer this gel over the nitrocellulose membrane, that is a nylon filter. Then after transferring your all the proteins that were present over the gel, they will be transferred over the nylon membrane, and then you probe them with the phosphorus 32p, which you could see on this like this one. It's analyzes that this I am present here. Then you expose to X-ray film, so all the others will be removed out, and you will see your corresponding result. Yeah. So there are 4 techniques: southern, northern, western. So in order to remember them, which techniques to which one, remember this short form: SNOW. Southern, northern, this is nothing, then western. And then remember rain. Was it rain or drop? Sorry, it's a drop. D R, snowdrop. So S for southern, N for northern, W for western. So D correspond to DNA, R correspond to RNA, and P corresponding to protein. So southern blotting is basically done to check the DNA, northern blotting is done to check your RNA, and your western blotting is done to check your protein parts mainly. So remember this concept. So southern blotting is the method for probing for the presence of specific DNA sequence within a DNA molecule. So DNA samples are separated by gel electrophoresis and then transferred to membrane by blotting via capillary action. And the membrane is then exposed to labeled DNA probe that has a complement based sequence, the sequence on the DNA of interest, and then less commonly used due to the capacity of other techniques such as PCR. So this technique is widely used when you want to make your transgenic mice, transgenic gene knockout mice, embryonic stem cell lines. So all these way it's required in all these things. So in order to identify the DNA, basically DNA allied applications. And northern, as we discussed, it's mainly done to detect the RNA, and the western is done to check the protein. The basic difference is that after transferring your protein over the nitrocellulose membrane, you have to run over the primary antibody and secondary antibody, and then you can check the results with the help of chemiluminescence, and then you can see the bands that you're looking for. So don't worry, I will have a one day dedicated to southern, northern, western. There is also western, northern, western also, which detects the combination of DNA and protein and RNA and protein. Yeah, there is a zoo blotting also, there's a dot blotting also. There is a one session for that, and with proper virtual videos about them also, and how we do all these techniques. And today our main focus is just to get to know about the various tools in the molecular biology here. Just to know the various tools in the molecular biology. Okay, we have good number of students, around 35. That's good. Slowly you will get used to it. It's just to get acclimatized. Yeah, acclimatize means like you go from one sphere of your comfort zone to another sphere, so you need to make yourself adjusted to new places, so things get different. So you might be not adjusted to 9 p.m. classes, so this needs some time in order to do so. So molecular markers, yeah, they are very big in the market. So Reliance company, Syngenta, Monsanto, all the big companies in the transgenic field of plants, in the GMOs, GMF, genetically modified food, genetically modified plants. So all these are being governed by the molecular markers, and they give you very quick results, like as a normal plant breeding which give you results in 10 years. With molecular markers you can get your results within 2 years. Your crop is ready with the specific traits that you're looking for, with the things that you want to make an impact on the society. So molecular markers, they are based on the naturally occurring polymorphism in DNA sequence. So genetic markers are sequence of DNA which have been traced to specific locations on the chromosomes and associated with the particular traits. It can be described as a variation that can be observed. A genetic marker may be a short DNA sequence, such as a sequence surrounding a single base pair, that is single nucleotide polymorphism, SNP, a long one, or like mini satellites. Yeah. So these are some various examples of genetic markers like Rflp, restriction fragment length polymorphism, AFLP, amplified fragment length polymorphism, RAPD, random amplifications of polymorphic DNA, VNTRs, variable number tandem repeats, microsatellites, polymorphism SSRs, SNPs, single nucleotide polymorphism, STR, short tandem repeats, SFP, single feature polymorphism, diversity array technology, and RAD markers. Then 5 conditions that characterize a suitable molecular markers. They should be polymorphic, codominant inheritance, randomly and frequently distributed throughout the genome, easy and cheap to detect, and reproducible. So molecular markers can be used for several different applications, including germplasm characterizations, genetic diagnostic, characterization of transformant, study of genome, organization and phylogenetic analysis, paternity testing and investigation of crimes, measure the genomic response to selection in the livestock. Then Rflp, these are the restriction fragment length polymorphism. They involves fragmenting a sample of DNA by restriction enzyme which can be recognized and cut DNA wherever specific short sequences are occurring. Yeah, so as the name suggested, restriction fragment, you restrict one fragment and then you polymorphize that part later on for the further genetic analysis. Then amplified fragment length polymorphism. In this we can amplify restricted fragments and can be used for comparison between your closely related species. So here we use a PCR technique actually. You increase the material to analyze it in better way. Then RAPD, these are the random amplification. You amplify a random part of your molecular markers and then you polymorphize that part. Yeah, so don't worry, just at the moment try to remember the full form at least. Yeah, so we will also have one lecture about molecular markers. I have added this part because this is very important. From the it's a future, these molecular markers, remember all these terms from the industrial perspective. In India they are the future, and there will be lots of job will be coming in agriculture with respect to them. It's gonna be huge market for it. Huh? Then microsatellites, SSR, simple sequence repeats. They are the microsatellites, simple sequence SSRs, or also called as short tandem repeats, STRs. They are the repeating sequences of 1 to 6 base pairs of DNA. Then comes SNP, a single nucleotide polymorphism. Is a DNA sequence. Variations occur when a single nucleotide like A, T, C or G in a genome that differs from the members of the species. So let's say you have A, T and A, and it got differentiated by the because of short nucleotide polymorphism into C and G, and because of that this it will give a new trait, could be used in the biomedical research, crop and livestock breeding further. Then these are STRs, they are the short random repeats in DNA, which occurs when a pattern of 2 or more nucleotides are repeated, and repeated sequence are directly adjacent to the each other. For example, CATG, it's being repeated, like it's around 4 base pairs here, and it's being repeated over the whole your sequence. And these cases are used in the forensic science, also in the genetic fingerprinting. Yeah, so if somebody is interested with the um, forensic science, that's also in the crime scene, people go to collect the DNA, yeah, and then they check whether it's from the victim who did that, and whether the DNA belongs to that person who was the culprit. So in that case your genetic fingerprinting plays a role. So DNA fingerprinting will be also be taught in detail. Now all in all we have discussed quite major molecular markers that are important in many perspectives. Now I will try to take you towards the most important part. If you want to start your um, yeah molecular biology, it's DNA isolation. Yeah, and then further you can do also RNA isolation, but DNA isolation is one of the important part. So we will learn basics things that has to be accompanied while doing this steps. This is your DNA isolation and how you can make the best out of it. So DNA can be isolated from any nucleated cell. So DNA is actually a giant anion in a solution. So sources of DNA includes, you can take DNA from blood, buccal cells in your mouth, cultured cells, bacteria, biopsies, yeah, forensic samples like body fluids, hair follicles, bones, and teeth roots. So DNA isolation is a routine procedure to collect DNA for subsequent molecular analysis. So there are 3 basic steps in DNA extraction, right? It's cell disruption, isolation of DNA, and precipitating the DNA. So in the cell disruption, this is commonly achieved by grinding or sonicating the sample. So removing membrane lipids by adding a detergent. Then isolation of DNA, it's removing proteins by adding a protease. And then precipitating the DNA, usually their ice cold ethanol or isopropanol is used. Since DNA is insoluble in these alcohols, it will aggregate together. So at the end you get a pellet upon the centrifugation, and this also remove all the alcohol that are soluble salt in it. So here we can see you have a cell lysis, you add FBAG reagent and protein degradation reagent, that is protein kinase K, into your sample. Then the binding happens, then you remove it, then the washing is done with the wash buffer, and then elution of your things, and then you have a pure genomic DNA. Then you have a pure genomic DNA, right? Then the basic rules, so blood first lies. The red blood cells with a gentle detergent such as Triton X100. Then wash cells as a hemoglobin, which inhibits friction enzymes and Taq polymerase, which works on ice to slow down enzymatic process. Then wears gloves to protect your samples from you, and autoclave all solutions stores in fridge. And keep all pellets and supernatant until you have the DNA that you are looking for. So how to get the DNA? So you take your cells, lyse all cells in presence of your sodium chloride NaCl, which helps actually to stabilize your DNA, and your DNA remains double helix because of NaCl. Yeah, and then comes your EDTA, which chelates your magnesium ions present in it, and as a cofactor DNAs which chews up the DNA rapidly. So it helps in chelating and removing the magnesium ions. Then anionic detergent SDS is added, sodium diadecyl sulfate, which removes the lipid layers. If any lipid layers are present in the cell, it helps to dissolve these membranes and binds positive charges of chromosomal proteins, that is your histones, to release the DNA into the solution. Because you know your DNA also have histones. If you go into the DNA, I don't know whether we have discussed that part. That we have discussed that you have a double helix DNA, but this double helix DNA is been coiled over the histone molecules, and these histone molecules are like a ball, yeah, and it's make a 1.5 turn over it, and then it's go further, and these balls together comes together and makes a like very tiny bunch of like a rope, and they are all concentrated into the chromosome at the end. So in that case, these DNA is revolving all around your histone molecule, yeah, and includes protease also, like protein kinase K, which helps into digest the proteins and incubate the solution at an elevated temperature for 4 to 24 hours. Then getting rid of the protein, so organic solvent extractions using equal volume phenol is to chloroform, so like 24:1, yeah. So you can use organic solvent extractions using equal volume phenol is to chloroform, that is 24:1. And then protein at the interference after the centrifugation for 10,000 rpm, that is round per minute, at 10 degrees Celsius for 10 minutes. So in this way you can remove the protein present in the sample. So you see, it's very there are so many things in the cells present, and we are very much focused on your DNA. So we don't want any RNA, we don't want any protein, we don't want any bilipid layers, we don't want any magnesium, yeah, we don't want any other salts in our sample. We don't want anything with the help of alcohol we were precipitating our DNA. We also don't want alcohol. Yeah, so remember that how all these things, it doesn't seems to be work in a single day. In order to get your DNA, somebody who might be working with this protocol, he might have to go through many hurdles and many things. Yeah. So my point here is why this line is coming up here. My point, yeah, histone ectomer. My point here is that in a research, in a protocol that you are reading at the moment, they are not what do you say, in a single day job. It's not a single day job. So there are a lot of troubleshooting, there are a lot of things that you see that this is working, this is not working, this might work, this might not work. So in order to do so, in order to find that, you have to keep working until you get success. So this protocol might have, just understand, the person who is working on this protocol saw that I have to just extract DNA, and there are many other things. So in order to save them, he added NaCl, he added EDTA, he added protein kinase K, yeah, he added phenol and chloroform ratio different, he added alcohol. So that at the end, that what you get is pure DNA. What you get at the end is pure DNA without any impurities of protein or RNA in it. So that's the focus about it. Okay, sir, but RBCs don't have nucleus. How we can extract DNA out of it? Okay, where RBC contains nucleus until the maturation. So DNA has to, after maturation, it destroys nucleus. Oh, thank you, Vaishnavi. Even I was not aware of that answer. That's very tricky and very good question, Pooja. I really like your question actually. Okay, good, good, good. Nice. Mm-hmm. So, yeah, yeah. Then you precipitate the DNA, then you add your 2.5 to 3 volumes of ice cold 95% ethanol and then leave it overnight at minus 20 degrees Celsius. Then you centrifuge sample at 10,000 rpm for 10 minutes, yeah, for 4 degrees Celsius. Then you wash your DNA pellet, remove excess salt in 70% ethanol and air dry. Then resuspend in sterile distilled water, pH 7.4. Then store at 4 degree or frozen at minus 20 degree Celsius on the long term. And quantifying the DNA, the amount of DNA can be quantified using this formula. That is, then in order to find out the DNA concentration, that is microgram per ml, you need to find out the optical density at 260 nanometer. Then you multiply with the dilution factor and then how much sample it is you present, that is 50 microgram per ml, then divide by 1000. So if the value is coming around 50 microgram per ml at the optical density of 260, then it's a double stranded RNA. If it is 33, then it's a single stranded DNA. Then if it is 40, it's called as, it's a RNA actually. Yeah. Single stranded RNA and double stranded DNA. And for the DNA purity, the purity of DNA is reflected in the optical density of 260 to 280. So ratio, and the ratio must be between your 1.6 to 2.0 if your DNA is pure. If it is less than 1.6, it means protein contamination is there. If it is more than 2.0, then it's a chloroform or phenol contamination. Then you need to re-purify your sample. So lots of patience, yeah, lots of determination, yeah, and full strength towards your goal that you want to achieve. Yeah, that's the 3 keys in order to make your protocol successful. So in summary, what we have done is, here we have the sample for DNA extraction, and then we lyse for cells at elevated temperature plus detergent plus enzyme in the salt buffer. Then we remove the cellular proteins, then precipitate of nucleic acid with ethanol, then quantify and purify your sample for the DNA. That's it. So that's it from this point of view, from this lectures, from the basics of molecular biology. Yeah. Now I will like to take you towards the practical aspect, how you can make how we can make DNA isolation. Good. Weather is good. Molecular biology is good. The protocols are good. DNA extraction is good. So let's start. I will stop talking now. Protocol ready to speak.
Before beginning any protocol, you want to make sure that your workspace has a proper flow to avoid contamination of samples or reagents. This example is set up for a right-handed student. Tips and pipe headers are on the left, racks and samples are in the middle, and the discard bucket is on the right. As your hand moves through the flow, it never unnecessarily crosses over the sample again. This video begins after students have spit into the DNA Genotech collection kits, capped the tubes to release the buffer, and mixed the solution by inversion. At this point, point, sample tubes should not be labeled to preserve student anonymity during the genotyping process. Using the P1000 micropipetter, 500 microliters is transferred from each spit collection tube into a new microcentrifuge tube. When pipetting large volumes, draw up the solution slowly to avoid bubbles or contaminating the micropipetter. Once the solution is transferred, close the spit tube lid and cap the sample. Repeat this process for all remaining samples. Prior to beginning this protocol, the heat block should be dialed to 50 degrees and turned on by flipping the switch. If it's heating, the light will glow red. Place samples in the heat block. This incubation must proceed for at least 90 minutes, but can also be left overnight. Tubes containing 20 microliters of DNA Genotek's proprietary purifying solution, Fragment, will be provided. Once the samples have been incubated, you will transfer the entire volume to a tube containing the purifying solution. You may notice that the solution in the tube becomes cloudy. This is normal. Mix the solution with a vortex. To turn on the vortex, press the switch down to touch and turn the knob clockwise to ensure maximum speed. Hold one tube at a time and press the top of the black pad to initiate vibration. Repeat this process for all remaining samples. Embed all samples in ice for 10 minutes. Impurities in the solution will be removed by centrifugation. After flipping the switch in the back of the microcentrifuge, press the open button. Remove the metal lid inside by pulling on the knob. Make sure that the tubes are labeled either with letters or numbers before loading them into the centrifuge. Load samples with hinges facing outward. And balance the tubes across the rotor by ensuring that all tubes have equal volumes. Replace the metal lid and press it until it clicks securely onto the rotor. Close the lid to the microcentrifuge gently. Change the centrifugation time by pressing the arrows to the desired time, in this case 5 minutes. Press the arrows by the speed to navigate to the proper RPM. In this case, 14,500 RPM is the max this centrifuge can perform and is close enough to our desired speed. Press the start button. The centrifuge will begin to spin. It is suggested that you watch the centrifuge until it reaches maximum speed to ensure that there are no violent vibrations, which may mean that the tubes are unbalanced or the internal lid has come loose. If this occurs, press the stop button immediately. As the spin is going, obtain 4 new tubes and label them. When the spin is complete, the lid to the centrifuge will pop open. Remove the internal lid and remove the samples gently to avoid disturbing the pellet. Place tubes of the same sample in line with each other to ease the transfer of the solution. After centrifugation, you should be able to see a white pellet that has collected on the bottom part of the tube on the side of the hinge. This pellet contains the impurities in the solution. The DNA is in the supernatant. Using the P1000, transfer 400 microliters of supernatant, which contains the DNA, to the new tube. Place the tip on the opposite side of the tube from the pellet and draw up slowly to avoid disturbing it. Repeat this process for the remaining samples.
Let's start the part 2.
Precipitate the DNA from solution by adding an equal volume of 100% ethanol to each sample . And mix by inversion 10 times. The solution may become cloudy. This is normal. After incubating at room temperature for 5 minutes, place the samples in the microcentrifuge with the hinges facing outward. Remember to balance your tubes. Spin the samples for 2 minutes. Remove the tubes from the centrifuge gently, being mindful not to disturb the pellet which now contains your DNA. Visually inspect for a pellet at the bottom of the tube on the side of the hinge. A pellet is not always visible, so you can choose to centrifuge again or proceed as normal. In this example, a pellet is only seen in samples C and D, but all samples are carried through the process. Using a P1000, slowly remove and discard approximately 800 microliters of supernatant. Place the tip at the bottom of the tube on the opposite side of the pellet to avoid disturbing it. It is not important at this point that all solution is removed. Just get as much as possible. Expel the tip with the solution inside into the discard bucket. Repeat this process for all remaining samples. Visually inspect the tube to see if this pellet is still present. If it has gone missing, make a note of that. Clean the DNA pellet by adding 250 microliters of 70% ethanol to each sample. Do not mix this solution, as it is imperative that the pellet is not disturbed. Repeat this process for all remaining samples. Place the samples in the centrifuge, being mindful of balance, and spin for 2 minutes. Remove the tubes from the centrifuge carefully. In this step, the pellet still contains the DNA, and it is important to remove as much supernatant as possible without disturbing the pellet. So remove and discard the supernatant using the P200 for precision. This will take 2 or more draws. On the first draw, you can expel the solution into the discard bucket and use the same tip to draw up again, as long as the tip has not touched other surfaces. Repeat this process for the remaining samples. Lay out a few layers of Kim wipes. Open the tube and knock it onto the Kim wipe to remove any large volumes of solution still remaining. Leave the tube open and lay it on its side to evaporate any of the remaining solution from the pellet. Repeat this process for all remaining samples and allow them to air dry for 20 to 30 minutes. If the tube still has spots of solution or if the pellet does not look dry, allow for evaporation to continue. If the pellet looks dry, then resuspend the DNA using water. Add 100 microliters of molecular biology grade water directly onto the pellet side. Cap the tube and mix by vortexing for a few minutes. Visually inspect the tube to ensure that the pellet has disappeared or has gone back into solution. If it has not, then continue to vortex or allow the tube to sit at room temperature before proceeding to subsequent steps. Repeat this for all remaining samples.
That was all about DNA isolation. I think I will not continue further here today as
Before beginning any protocol, you want to make sure that your workspace has a proper flow to avoid contamination of samples or reagents. This example is set up for a right-handed student.
because I want to start with the new
Tips
topic
and
with a
pipetters
fresh
are on
start,
the left,
so it will not make sense.
racks and samples are in the middle, and the discard bucket is on the right.
It has both parts,
As your
part
hand
1 and
moves through
part
the
2.
flow, it
So you can
never
watch them from
unnecessarily crosses over the sample.
So that's it for today. So what's the time? Still we have 7 minutes, but relax students. I don't want to make it so hard and fast for all of you. So thank you very much. So I hope you had learned the basics of molecular biology with me on these first 2 days. A little bit about cell culture, transcription, translation, central dogma, various tools in molecular biology, gel electrophoresis, molecular markers, PCR, DNA isolation. Yeah, so we went through quite number of things within the short span of time. Yeah, my main aspect is here that we can grab as many as things within the short span of time, and You can have all these things with yourself also, because some teachers tend to do what, they make these presentations and they do not like to share with their students. I've seen many in my career also. But my reason is here, if I keep knowledge with me, if I keep my all aspect that I'm sharing with my students with myself only, then my growth will be decreased, might be my growth will be somehow will stop if I do not share things. So all the presentations that I have shared with you, go through them, try to make assignments out of it, yeah, and yeah, start working on a review article, because if you get late, then your certificate will be late then, yeah. So on that note, thank you very much everyone for cooperating with me. So we will see us again. Today is Wednesday, or yeah, today is Wednesday. So we see us again tomorrow at 5 p.m. Sharp. At I will share link 10 minutes or 5 minutes before, and we will start the lectures at 5 and continue for further things. Yeah. So thank you very much. Wish you all good night. Have a nice sleep. Yeah. Maybe these techniques coming into your dreams. That's good. That you're the future molecular biologist. You never know, and one of the Nobel Prize winner also might be. So that note, take care. Bye bye. Sayonara. Good night. Ciao.